that you have no essential textbook for you to prepare, I decided to make you this lecture with explanation of the topic for you to be more prepared for your first topic. You should buy the album. It will have yellow color. If you didn't buy the album, please do it as fast as you can. So you should come to your first class, practical class, with your albums. You can fill the album in the first topic if you know how to do it, or you can just come with your album and then teacher will learn you what to do there. But now I will give you some theory about the material. So your first topic will be cardiovascular system. First, about the development, embryonic development. In embryogenesis, blood vessels and also the cells of blood-formed element arise from mesenchyme that derives from visceral layer of lateral plate mesoderm. And first blood vessels appear in the yolk sac. If you remember, what is it? This is the section through the embryo. Here is the embryo itself. Embryo is covered with amnion, and below the amnion, below the embryo, you can see yolk sac. And here in the wall of the yolk sac, in the mesenchyme, you can see the blood islets, in which the mesenchyme cells turn into either primary erythroblasts or blood cells, or into endothelial cells. Endothelial cells are the cells of blood vessels. So the first blood vessels are formed outside the embryo, in the yolk sac, then in the chorion, and only after it in the embryo body itself. And in the embryo body, as you remember on this preparation, the blood vessels appear from the mesenchyme derived from visceral layer of lateral plate mesoderm. This is the visceral layer of lateral plate mesoderm. Here is the endoderm, and between these layers there is the mesenchyme, from which blood vessels are formed. Now, our system, cardiovascular system, includes heart, blood vessels, and lymphatic vessels. Let's talk first about the blood vessels. Blood vessels include large and medium vessels. They are arteries and veins, and small vessels that are called microcircular bed. Microcircular bed includes arterioles, venules, and capillaries between them. Or instead of capillaries, blood can flow through the arteriovenous anastomosis. We shall discuss them later. Now, this is important information. Important information means that you should learn this by heart. Blood vessels, the sequence of blood flow. You should remember through which vessels, in histological sense, the blood flows from heart. From heart, first blood enters the elastic artery, then into mixed arteries, then into muscular arteries, then into arterioles, then metarterioles, or they are also called precapillaries, then into capillaries or arteriovenous anastomosis. If blood goes into capillaries, the blood changes from arterioles into venous. It means it lacks oxygen and becomes poorly oxygenated. Or if the blood goes directly from metarteriole into venule through the anastomosis, it is still red in color. It is high highly oxygenated. Then venules. Venules, they include postcapillary venules, the smallest of them, collecting next ones, and muscular, the largest venules. Then small and medium veins, which can be either muscular or fibrous. Then into large veins, for example, vena cava or pulmonary veins, and into heart. So you should know this by heart, types of blood vessels in their sequence. Now, this is also important information that you should know by heart, the general structure of the blood vessel wall. All blood vessels have their general structural plan that includes 3 layers or tunics. Tunica interna, which is also called tunica intima. Middle one, medium layer, is called tunica media, and outer layer is called tunica externa or adventitia. We usually use the terms intima, media, and adventitia for these 3 layers of the vessel wall. Now, each layer consists of sublayers. Tunica interna is the innermost layer, and it includes endothelium. Endothelium is always the innermost layer in any vessel, and also inside the heart. Heart is also lined from inside with endothelium. Endothelium is the layer of flat cells. In red color, I added here the information about the functions. So the functions of endothelium is the transportation of gases, nutrients from blood to the underlying tissues. Also, it provides clot formation, or in normal state, it prevents clot formation. Also, endothelial cells secrete some molecules, and endothelial cells provide laminar flow of blood. What does it mean? It means that, as you remember, endothelial cells are covered with glycocalyx, which is negatively charged, and also formed elements of blood are negatively charged. So formed elements of blood do not stick into endothelium, do not stick to the wall of blood. So endothelium provides laminar blood flow. Next, subendothelial layer is a layer of loose connective tissue, loose fibrous connective tissue. It provides protection, shock absorption during the pulse wave, and also nutrition of underlying tissues inside the blood vessel wall. And outermost layer of interna is internal elastic membrane. It's a membrane consisting of elastic fibers, and it prevents overstretching of the wall or collapse of the lumen, or it prevents closure of the lumen of the wall of the vessel. Lumen means the internal space of any whole organ, and also the inner space of any blood vessel. So internal elastic membrane is elastic because it consists of elastic fibers, so it can stretch and return into its normal state. So it prevents overstretching and rupture of the blood vessel, and also it prevents closure of the lumen of the vessel. Next, the medium tunic, the tunica media consists of smooth muscle cells and some elastic components. These elastic components are produced by smooth muscle cells. As you remember, smooth muscle cells in smooth muscle tissue can produce components of extracellular matrix, including elastic fibers. This tunica media provides muscles, provides support of the wall of the blood vessel, vasomotion, which includes constriction and dilation. It means that smooth muscles can contract and provide vasoconstriction, or muscle cells can return to their normal state and relax and cause vasodilation, increasing of the lumen of the vessel. Also, smooth muscles regulate the blood pressure inside the vessel because they can contract. When the smooth muscle in the vessel contract, the blood pressure increases. When the tunica media with smooth muscles relaxes, then blood pressure drops. It's about the smooth muscle cells in tunica media. What about elastic elements? Elastic elements provide elasticity. It means that they can stretch and recoil again, preventing both overstretching and rupture and collapse of the lumen of the vessel. And outermost tunica externa or adventitia. It includes external elastic membrane. It resembles the internal elastic membrane. It also consists of elastic fibers and prevents overstretching and collapse of the lumen. And the outermost layer is the loose connective tissue with vasa vasorum and nervi vasorum. Vasa vasorum means vessels that are located inside the wall of some very large vessel and provide blood supply of the tissues and cells of these large vessels. So each vessel consists of tissues and cells. Each of them wants to be supplied with oxygen and nutrients. And if the vessel is very large, it should have some additional blood vessels supply with oxygen and nutrients the cells. I'm sorry, I was kicked out from my conference, but I'm now here again. So we were talking about the adventitia, in which loose connective tissue provides protection of the vessel, binding the vessel to the surrounding tissues, flexibility of this vessel, because vessels are located in our body, and when the body moves, the vessels should have some flexibility to not be ruptured. Nutrition and innervation, because of the presence of vasa vasorum and nervi vasorum, blood vessels and nerves that supply the wall of this vessel. This is the general structural plane of blood vessels, and in different blood vessels, we will have somewhat different structure. This is the schematic drawing of previously discussed structure of the blood vessel wall. So the innermost layer of flat cells, endothelium. Endothelium lies upon its basement membrane, and under it we can see loose connective tissue and internal elastic lamina. This is intima. Next, middle layer is called media, tunica media. It consists of smooth muscle cells and some elastic elements, and also some amount of collagen fibers, other fibers, amorphous substance, and very tiny amount of loose connective tissue. And outermost layer is adventitia, which includes external elastic lamina or membrane and loose connective tissue. And here we can see the blood and nerve supply. These will be called vasa vasorum and these nervi vasorum. They come inside the wall of this vessel and innervate and supply with nutrients and oxygen the cells which comprise the wall of this vessel. So, all the blood vessels have the same general structural plan of their walls, which includes 3 layers. But why the vessels differ in their microanatomy of the wall? First, because of hemodynamic conditions. Hemo means blood, dynamic means how this blood flows. Which are these hemodynamic conditions? They are speed of blood flow, how fast blood flows, the pressure, high or low, is it pulsatile or continuous, and gravity also may play the role. And second, functions. So the blood vessels differ in their histology because of first hemodynamic conditions and second functions. Let's look at different types of the vessels in this sense. For example, arteries. What hemodynamic conditions we have in arteries? We have high speed of blood flow, speed is high, pressure is also high because the blood comes from heart, and also the flow is pulsatile because of the heart. Gravity doesn't play a role here. What are the functions of arteries? Arteries should resist this very high blood pressure and blood flow. Arteries should deliver blood at long distances to the organs and tissues of our body without losing of the blood pressure, which is made by heart. Heart makes the initial speed and pressure of blood. And also arteries regulate the systemic blood pressure. So this is why arteries have thick wall to resist blood pressure. They never collapse because they should deliver blood at long distances. They cannot be collapsed with closed lumen. They have relatively thick media, and in this media they have a very well developed muscle tissue and elastic elements. Well developed means that in histological section we will see a lot of such elements. Well developed means we see many of something. We will see many muscle cells. We will see many elastic elements. Muscle cells provide resistance to blood pressure and flow. They also can regulate blood pressure. If muscles contract, blood pressure increases. If muscles relax, blood pressure decreases. Elastic elements provide resistance to blood pressure, and they prevent collapse of the lumen. Veins return blood towards heart. What are the hemodynamic conditions in veins? In veins, the speed of blood flow is very low, and also the blood pressure is very low. In large veins, such as vena cava, which drains into the heart, we have blood pressure about 0. No blood pressure at all. So the blood pressure is very low. The blood flow is not pulsatile, it is continuous. Almost every time it is continuous. And in some cases, gravity plays its role. For example, if we are talking about the veins that are located in our legs and other parts, lower parts of our body, where the veins should move the blood from our lower parts up to the heart. It means that the blood goes against gravity. Functions of veins. They provide outflow of blood from organs, they return blood to heart, and also they store blood. About 64% of all our blood every moment is located in our veins, not in heart, not in arteries, not in capillaries, but in veins. Veins store the blood. So veins have thin wall because of low speed and low blood pressure. They should not resist high pressure. They have relatively thin media with poorly developed muscle tissue and poorly developed elastic elements. They may collapse, so in our body, veins can be closed with closed lumen, but arteries cannot be closed. And also, veins may have valves. All the veins of our lower part of our body have the valves. Valves prevent backflow of blood. Capillaries. In capillaries, we have low speed and low blood pressure, and the blood flow is continuous without pulsing. Functions. Capillaries provide gas exchange and exchange of nutrients and metabolites between the blood and tissues. And also capillaries provide outflow of fluids from tissues. So capillaries are the only vessels for gas exchange. Gas exchange can be promoted only by capillaries. All other types of blood vessels have too thick wall to provide gas exchange. So gas exchange is only through capillaries. And this is true also about nutrients and metabolites. So the capillaries have very, very thin, permeable, and fragile wall. Arterioles are actually the smallest arteries. What are their hemodynamic conditions? The blood flow slows down in arterioles because the blood flow is very high in arteries and very low in capillaries. So in arterioles, which are in between, the blood flow slows down. The same as about the blood pressure. Blood pressure drops. Blood pressure is high in artery and very low in capillary. So in arterial, it drops down. And the blood flow changes from pulsatile to continuous because the capillaries have very thin wall and they cannot resist high pressure and pulsatile blood flow. Functions. Arterioles regulate the blood pressure. They are the main blood vessels in our body that regulate the blood pressure. They regulate the blood delivery within organs. It means that arteriole can decide if the blood will flow into, for example, this capillary or this one, because arterial may close or open its lumen. If the lumen of the arterial between the arterial and capillary will be open, so the blood will flow into this capillary. If the arterial closes its lumen, then the blood will not go into this blood capillary, it will go to some other. So this is how the regulation of blood delivery within organs happens. For example, in any moment, we do not use all the parts of our body. For example, we do not use all the muscles. It means that in our arm, for example, only one portion of muscles will be active, and only those capillaries will have the blood inside them, because of the arterioles that will close their lumen near the muscles that are not active in this moment. So this is the regulation of blood delivery. And arterioles finally make the blood flow suitable for capillaries. It means that they slow down blood pressure and blood flow, and makes the blood flow from pulsatile into continuous. So this is why the arterioles, their wall resembles that in artery. It has a well-developed muscle layer, one layer of smooth muscle cells in media, well-developed inner elastic membrane that prevents its collapsing, and metarterioles have sphincters. Metarteriole is the small arteriole between arteriole itself and the capillary. So metarteriole is just before the capillary. Venules resemble small veins and also capillaries. We have three types of venules. The smallest ones are postcapillary venules. They resemble capillaries in their structure. Then we have collecting venules, and finally muscular venules, which drain then into veins. So in all venules, hemodynamic conditions are low speed of blood flow, low blood pressure, and the blood flow is continuous. Functions. Venules provide the outflow of fluids from tissues. They also collect and store blood. And we have some specific type of venules, which are called high endothelial venules. High endothelial postcapillary, to be sure, venules. They are very specific venules which are located inside the immune organs, such as lymph nodes and spleen, which provide lymphocyte trafficking. Through the wall of such venules, lymphocytes can enter the blood vessel or go out from such venule. They are very specific in thymus and in lymph nodes. We can find such venules. So venules have a very thin and permeable wall, which resembles that in veins. Now, very important information, the classification of arteries, which you should know by heart. Arteries can be divided into elastic, muscular, and transitional type, mixed type. This classification is based upon the ratio of muscle and elastic elements in the media of these arteries. If the muscle cells prevail in volume, then this artery will be called muscular. Tunica media is predominantly composed of muscular elements. If the elastic elements prevail, it will be called elastic. And we have only two elastic arteries in our body. They are aorta and pulmonary trunk, which go out from heart. Only those two are elastic arteries. And in these arteries, the elastic elements are organized in so-called fenestrated laminae or fenestrated membranes. So these are not the fibers itself, but the membranes, which are cylindrical and are located one inside another one. Smooth muscle cells are also present in elastic arteries, but elastic elements prevail. And if we have the equal amount of muscle and elastic elements, we will have the mixed arteries. In our body, these are, for example, the arteries that go from aorta. Next, the branches of aorta will be, in histological sense, mixed type of arteries. All the other arteries in our body are of muscular type. We will see many sections of many organs, and in every organ we will have only muscular type of arteries. Elastic and mixed ones are aorta or pulmonary artery and their first branches, which are mixed ones. All other in our body are muscular arteries. So here in this picture, you can see the structure of muscular and elastic artery. What are the differences between them? First, elastic artery has more elastic elements than smooth muscles in media. Muscular artery, vice versa, which means opposite. So in elastic artery, this is intima, this is media, and this is adventitia. And in media, we have a lot of these wavy lines here. These are actually sections through the fenestrated elastic membranes. One membrane, another one, another one, another one. Between them, we will see smooth muscles. They are located between these membranes. So smooth muscles are sandwiched between membranes. In muscular artery, we have no elastic membranes. We can see a lot of smooth muscle cells that are circularly located. Circularly. Between them, there will be some amount of elastic fibers and other components of extracellular matrix. Second difference, elastic artery has fenestrated elastic membranes. Elastic membranes are present in elastic artery, no membranes elastic in muscular artery. And third one, muscular artery has internal and external elastic membranes. Internal and external elastic membranes. Only in muscular artery, no such in elastic one. Yeah, I'm sorry about that. So now every 40 minutes we will have a break about 10 minutes. It's Zoom. I cannot do anything about this. It wants break or money. Okay, let's look at the histological preparation. If you can hear me, I think you can. Okay, histological preparation of the wall of elastic artery. Elastic artery has a lot of fenestrated elastic membranes in its media. So here should be the lumen of this artery, innermost layer, tunica interna or intima, which includes endothelium and thin underlying loose connective tissue, which is called subendothelial layer. It is under the endothelium, and here starts the media, which includes fenestrated elastic laminae. They look here like light waves, and between them we should see smooth muscle cells. So in tunica intima is here. From this point to this point is tunica media, and outermost tunica externa or adventitia. It's loose connective tissue with blood vessels and nerves. Blood vessels are called here vasa vasorum, and nerves will be called nervi vasorum. So these blood vessels feed the cells and tissues of the wall of aorta. Another example of this preparation, elastic artery, aorta, cross-sected with Orcine staining. If you remember, Orcine is for elastic fibers. So here, reddish brown are the stained with Orcine fenestrated membranes. Between them, we can see the cell nuclei of smooth muscle cells, smooth muscle cell nuclei, and fenestrated elastic membranes. This is the tunica media, the thickest. Tunica intima, innermost, and tunica adventitia, the outermost one. In our class, we should look onto our histological preparations. This is our specimen, aorta in the cross-section stained with Orcine. We shall discuss this preparation in our class, but now I will show you the general structure. The intima, thin, media, thick, and adventitia. The higher magnification, innermost tunica intima, and part of the tunica media with stained fenestrated elastic membranes. Between them should be smooth muscle cells that are not seen here because of Orcine staining. It doesn't stain nuclei. Another preparation which you should draw in your albums, and it will be discussed in class, aorta on longitudinal section, stained with hematoxylin eosin at low magnification. Tunica intima, tunica media, very thick, and tunica adventitia with blood vessel, nerve, and this is the section of the first branch of aorta. So this will be the section through the artery of mixed type. Aorta itself is the artery of elastic type, and this one is mixed artery. Okay, important information. You should know this by heart. Classification of veins. Veins are divided into muscular and fibrous or amuscular, depending on the amount of muscle tissue in the wall. If the vein has no muscle tissue in the wall, it will be called fibrous or amuscular. A means no muscles in the wall. These are the veins of the spleen, bones, placenta, pia mater, retina, and some other. All other veins are of muscular type, and they are subdivided into 3 subtypes based, again, upon the amount of muscle tissue, with low, medium, or highly developed muscular tissue. Muscular veins with prominent muscular elements are located in the lower part of our body because they should provide blood flow towards the heart from our low parts of our body towards the heart. In our upper part, we will have muscular veins with low developed muscular elements, and in the middle part of our body with medium developed muscular elements. Here is the drawing of comparison between muscular artery and muscular vein. Look at the thickness of the wall of muscular artery, wall of the vein, and the thickness of the tunics. Tunica media in artery is thicker than tunica adventitia, and vice versa. In a vein, the tunica media is thinner than adventitia. And look at the well-seen internal and external elastic lamina in muscular artery. We cannot see them in vein, but the general structure is the same in artery and in the vein. Tunica intima in both of them, tunica media with a circularly located smooth muscle cells, and tunica adventitia with loose connective tissue. This is how the muscular artery and vein look like. Tunica intima, tunica media with elongated cell nuclei of smooth muscle cells in tunica media, and tunica adventitia, which consists of loose connective tissue, and these are the cell nuclei of cells of loose connective tissue. The same is for the vein. Intima is very, very thin. Media includes elongated spiral-looking nuclei of smooth muscle cells, and adventitia with loose fibrous connective tissue. Now, important information. You should know the differences between an artery and a vein. You should be able to differentiate between them. So, what are these differences? First, artery has a thicker wall than a vein. Compare the thickness of the wall of the artery and the vein. Second, an artery has a more prominent media than a vein. Compare the media in artery and media in a vein. Media is much thicker in artery. Number 3, in artery, media is thicker than adventitia, and in vein, vice versa, media is thinner than adventitia. Compare thickness of media and of adventitia. In artery, media is thicker than adventitia, and in vein, media is thinner than adventitia. Number 4, an artery has a well-seen internal elastic lamina. It is highlighted here in green, and this is why the inner surface of the artery looks wavy, with waves. This is because of internal elastic membrane or internal elastic lamina. And in vein, we can see the flat surface without any waves, because in veins we have no elastic lamina. And also vein may have valves that prevent backflow of blood, but not in this histological picture. But you should know all these differences between arteries and veins. So, in this histological preparation, intima, media, adventitia, let's look once again at the internal elastic lamina, which looks here like a wavy pink line. Why it is so wavy? Because of smooth muscle cells of the media located outside of the internal elastic membrane. This is actually how the artery looks like when it is constricted. When it is constricted, its internal elastic membrane will be such wavy. It is so in our histological preparations because when we took some tissue and fix it in formalin or ethanol or some other fixatives, smooth muscle tissue and smooth muscle cells in it always contract during the histological preparation of a specimen. This is why all the smooth muscle cells are contracted and the smooth muscle tissue contracts the underlying internal elastic lamina. This is why it becomes so contracted, so wavy looking. But in our body, in normal state, arteries contract only when they want to, for example, to increase blood pressure. Also, in our body, arteries do not close their lumen. Their lumen is always open. They never collapse, but veins can collapse in our body. In histological preparations, both of them can look collapsed in some cases. Here, compare the vein and the artery, compare the thickness, overall thickness of the wall here and here, compare the thickness of the tunica media with muscle tissue in artery and in a vein, and comparative thickness of media and adventitia. Media is thicker than adventitia in artery, and media is thinner than adventitia in a vein. Here the internal elastic membrane is not well seen in artery, so we cannot use this how this is developed. The same is in this picture. We can compare the overall thickness of the wall of the artery and of the vein. Vein has a very thin wall comparative to artery. The same as here, small vein, small artery. These are endothelial cells that bulge into the lumen of the artery because of the internal elastic membrane that pushes them towards the lumen of the artery. Important information, you should be able to differentiate the blood vessels. So it means that you should name properly the vessel and you should explain why do you think that this is, for example, the artery. So you say, this is the artery. The question, why this is the artery? So you should be able to explain why it is the artery. Your answer should be like this. It's artery because it has a thick wall. Its wall is thick. If we compare it with the lumen, in the lumen we can see the erythrocytes. Second, its media is thicker than adventitia. Media containing smooth muscle cells is thicker than adventitia. And 3, it has a well-developed internal elastic lamina, pinkish wavy line here. And also you can be asked, is it an elastic or muscular artery? And you should answer that this is the muscular one, not elastic or mixed, because it is located inside an organ, it is rather small, and it has an internal elastic lamina. in elastic artery, we have no internal and external elastic membrane. And also it's in its media, smooth muscle cells predominate, and we cannot see any membranes here. The same is for a vein. So why this is a vein? Because it has a thin wall, because its media is thinner than adventitia. We almost cannot see here the media properly, only some muscle cells here. And it has no internal elastic lamina and, as a sequence, flat internal surface, not wavy. Another example of a small artery and a preparation of a vein. It's a vein of muscular type with highly developed muscular tissue. In such veins, this one is located in our legs. We have smooth muscle cells not only in the tunica media but also in tunica interna, intima, and adventitia. Adventitia is the thickest one here, but it also contains smooth muscle cells. In tunica media, in all the vessels, the smooth muscle cells are located circularly, but when we have smooth muscles in intima or adventitia, there smooth muscles are located longitudinally along the axis of the blood vessel. It's how it looks like in a high magnification: tunica intima, tunica media, and tunica adventitia with cross-sected bundles of smooth muscle cells. And this one is the vasa vasorum. Now, neurovascular bundle. In our body, artery and a vein always go together. So if you see the artery, then you should look around and see also its companion vein. Also, they usually have their third friend, a lymphatic vessel, and also a nerve fiber, or a nerve or bundle of nerve fibers. We have such preparation, a section, cross-section of the neurovascular bundle, in which you should define the artery. This is the artery, this is the vein, and these are bundles of nerve fibers. This will be your preparation, which you will discuss in class. On a high magnification, the cross-section of the artery of muscular type, its lumen, intima, consisting of endothelium and a thin subendothelial layer, and internal elastic lamina here, thick media, and thinner adventitia. Another example of this preparation: endothelium, subendothelial layer, and internal elastic lamina comprise intima, then media with smooth muscle cells and some fibers between them, and outermost adventitia, loose connective tissue. The same preparation on the highest magnification, endothelium. Here we can see a portion of subendothelial layer, loose connective tissue, and internal elastic membrane, tunica media, tunica adventitia. A vein has smooth internal surface because it lacks internal elastic membrane and also external one. And its adventitia, which is seen here better, is thicker than media. Tunica media with smooth muscle cells is thinner than adventitia. Overall thickness of the wall of the vein is not so thick as it is in artery, so the vein cannot hold always its lumen open. So we can often see the lumen collapsed in veins. Another example of a vein which has valves. Valves includes two cusps, one here, another here, which are duplicates of tunica intima, and they actually are pockets. One pocket here and another one here. Valves prevent backflow of blood. Example of small artery, example of small vein. Compare the thickness of their wall and amount of smooth muscles in tunica media, here and here. Another example of the vein with valves. In this picture, this is the section of the wall of the stomach. In its tunica submucosa, you can see a lot of blood vessels at low magnification. And here you can compare the thickness of their wall. If you compare this vessel to this one, you can compare the thickness of the wall, and you can say that this one is a vein and this one is the artery. Artery, vein. The same on a high magnification, and you will discuss it in class. The artery, and here the section plane is transversely. This is the cross section of the artery, and here is the same artery but sected through its tunica media. So this is why we can see here circularly located smooth muscle cells. Next, let's talk about microcirculatory bed, which includes arteriole, venule, and what is between them. So what can be between them? Usually it's capillaries, then from arteriole will start the metarteriole. The usual sequence of blood flow will be arteriole, metarteriole, or precapillary, then capillary, and venule, and then vein. Precapillaries or metarterioles have the sphincters that allow or not allow blood to flow into these capillaries, and thus regulating the blood flow inside the organ. But in some cases, some other cases, we have differences. For example, the blood can flow directly from arteriole into the venule through the arteriovenous anastomosis. Or we can have the portal system, where the blood capillaries are located between two arterioles, like here, or between two venules, like here. Venule here, and between them capillaries. This can happen in some cases when we are not talking about gas exchange and supply of tissues with nutrients and some other functions of blood capillaries, which provide this usual system with capillaries. For example, the capillaries between two arterioles can be found in the kidneys, in nephrons, where these capillaries are not for blood supply of tissues, but for production of urine from the blood plasma. The same as about the capillaries located between two venules. We can find such system in the hypothalamus, and in this case, these capillaries are also not for blood supply of tissues, but for delivery of hormones from point A to point B. We will discuss it in details on the topic about endocrine system. The histological structure of the blood vessels of microcirculatory bed. Arteriole resembles small artery. It includes endothelium with subendothelial layer and internal elastic lamina. in intima, and well-developed smooth muscle cells in media. Meta-arteriole has precapillary sphincters that can contract or relax. When they relax, blood will flow into these capillaries. When they contract, blood will not flow into these capillaries. Then, capillaries have the endothelial layer lying upon the basement membrane and scattered pericytes, and also adventitial cells may be present. Then venules, smallest ones are called postcapillary, and they resemble capillaries in their structure. So they also consist only of one layer of endothelial cells and some scattered pericytes. Then larger venules will have also smooth muscle cells in media and adventitia. So, important information. You should know the histological structure of blood vessel wall of arterioles, venules, and capillaries. The tiniest vessel is the capillary. It can have only endothelium in some cases. So some capillaries have only endothelium, but others can have also pericytes and adventitial cells. This is about capillary. It's the tiniest for gas exchange and some other exchanges. Venules. The smallest ones have only endothelium and pericytes, the smallest ones, but other larger ones can have also media with sparse smooth muscle cells and adventitia with loose connective tissue. And finally, arteriole resembles the artery in its structure. It always has three well-developed layers: intima, media, and adventitia. Intima contains endothelium, subendothelial layer, and always internal elastic lamina. And media contains one layer of densely packed smooth muscle cells, circularly located. Adventitia, as always, contains loose connective tissue. This is how different blood vessels look like on the sections. This is the cross-section of the artery with thick wall and wavy inner surface. This is the section of the vein, thin wall, smooth inner surface. And here some examples of small blood vessels of microcirculatory bed. Arteriole resembles artery, but it has only one layer of smooth muscle cells in its media. Venule resembles a vein. It also has very thin wall, and capillaries are the smallest ones. Their diameter may be equal to a red blood cell. It's about 7 micrometers or even smaller. We will discuss microcirculatory bed in class using our preparation of the pia mater. Pia mater covers the brain, and this is the whole mount preparation or total preparation. It means that we take the pia mater. It looks like a thin membrane. We stretch it upon the glass. We stain it, and finally, what we can see here, we look at the blood vessels in the pia mater, and we see them from above. So they are, we see them as a whole. So these are not the sections. Here, the whole blood vessel is seen, squeezed between two glasses and stained. And here you should be able to define arteriole, venule, and a capillary. This is how the arteriole looks like. It is branchy here. It's the only blood vessel here that contains circularly located smooth muscle cells, and they look like cross striation here. Nuclei of smooth muscle cells. If the smooth muscles are located on the lower or upper side of the wall of this vessel, their nuclei look like elongated, transversely located lines. If the smooth muscle is located on the side of the wall of this vessel, its nucleus will look like a dot. So these are also the smooth muscle cell nuclei, but they are located on the side of this wall. Elongated cell nuclei that are longitudinally located along the axis of the blood vessels are always endothelial cells. These are endothelial cells. In venule, we can see only longitudinally located, elongated endothelial cells and sometimes pericytes. The capillary is the smallest one, and its lumen is equal to 1 erythrocyte. Arteriole, endothelium, elongated along the long axis. Cross striation because of cell nuclei of smooth muscle cells, and these are also smooth muscle cell nuclei. A venule that consists of endothelium and some pericytes. Arteriole, venule, and a capillary. A capillary once again with endothelial cells and pericytes. And some adventitial cells can also be present. So, important information. You should be able to name the blood vessel and explain why do you think so. For example, why this is the capillary? Your answer. Because first, its wall has only endothelium, so these are cell nuclei of endothelial cells, but also pericytes and adventitial cells may be present. And the diameter of its lumen is equal to 1 erythrocyte. So this is why we can see 1 row of erythrocytes in its lumen. Not 2, not 3, only 1 row of erythrocytes. The same as about this one. This is the venule. It's branching here. It's a venule because in its wall we can see only endothelial cells, no smooth muscle cells, only endothelium, but also pericytes may be present. And the diameter of its lumen is more than 1 erythrocyte, so we can see several rows of erythrocytes in its lumen. Why this is the arteriole? It's the simplest question because it has cross striation because of circularly oriented smooth muscle cells. If you see cross striation, this is the arteriole. These are the pictures taken from the transmissional or scanning electron microscopes. In transmissional electron microscope photograph, we can see the cross-sected arteriole. Here, in its lumen, we can see 2 platelets and an erythrocyte. The innermost layer is endothelium. Endothelial cells are somewhat bulged into the lumen of the arteriole because of these very well developed internal elastic lamina and smooth muscle cells. So the same for this picture. Why this is the arteriole? Because of well-developed internal elastic lamina and smooth muscle cells that contract and thus push this internal elastic lamina, which in turn pushes the endothelium. This is the cross-section of the nerve fiber bundle with myelinated here and non-myelinated nerve fibers here. And this is the scanning electron microscopy photograph in which we can see the smooth muscle cells circularly located. And this arteriole is innervated by the sympathetic nervous system fiber, which makes this arteriole to contract or relax. Another example of the arteriole, endothelial cells. Here should be the internal elastic membrane and smooth muscle cell in media. And here should be the adventitia. Important information. Types of blood capillaries. We have 3 types of blood capillaries. This classification is based upon the absence or presence of pores or gaps or some thin areas in the wall of the blood capillary. The most continuous one, without any pores, any gaps, is the first one, which is called somatic, or a capillary with continuous endothelial cell. No pores, no holes, no gaps. It is continuous. It is covered with basement membrane. It is also continuous. And also pericyte and adventitial cell is present. Pericyte is covered with basement membrane from all the sides. And an axon of the sympathetic nervous system is innervating pericyte, making it to contract, to make the this capillary to contract, to make its lumen smaller. Location. Somatic capillaries are located in muscles, skin, lungs, brain. Their functions are to perform gas and nutrients exchange, and also they usually are the part of blood tissue barriers. For example, of the blood-brain barrier inside the brain, because it's very continuous with no gaps or holes. So it protects the tissue from something potentially bad, dangerous, harmful from blood. Second one, visceral one, has the so-called fenestrations or fenestrae. So another name is fenestrated. These are the areas with thin cytoplasm, and it allows some large amount of large molecules to easily pass through the wall of such capillary inside its lumen. And these are located in intestines, glands, endocrine glands, and kidney. And they transport large amount of molecules through their walls, through this area of more thin cytoplasm of endothelium. So we will have another break, about 10 minutes.
Okay, thank you.
Okay, let's continue while we are waiting for others. So we have stopped on the visceral type of the capillaries. They are also called fenestrated because of these fenestrations, where the inner and outer cytolemmas, cell membranes, come very close together, making the thickness of the cytoplasm of the endothelial cell in this place very thin, allowing very large amounts of large molecules to pass in this site through the wall of this capillary. So this is why such capillaries are located in places like intestines, where a large amount of some ingested food should enter the blood. Also in endocrine glands, where gland cells produce hormones that should enter the blood vessels, and in kidneys and in some other places to transport large amounts of large molecules through the wall. And finally, the third one type, sinusoidal capillary, the most discontinuous one, because it has pores and gaps in the endothelial layer and in the basement membrane. Basement membrane is also incontinuous, non-continuous, with holes in it. And no pericytes and adventitial cells are seen in this case. Where we need such capillaries with holes, in some places where we need not only large molecules but also the whole cells to pass through their walls. For example, in bone marrow we have sinusoidal capillaries, and in bone marrow the newborn formed elements of blood should enter the blood flow, and they enter the sinusoidal capillaries through such holes in their wall. And in liver, a large amount of large molecules also can pass through these holes, gaps and pores. This is how the pericytes look like, covering the endothelial layer. So this is the pericyte with its processes that hug the endothelial layer. And we have these electronograms in the colloquium, so you should be able to describe them. So you should name the blood vessels that you see here and explain. So this is the somatic capillary. Why? Because first, it's a small blood vessel, it's not artery or vein, it's a capillary, it is continuous. We can see no holes, no gaps, no pores in its cytoplasm. It has basement membrane that is also continuous with no pores and gaps. And here you can see a portion of a cut-off cytoplasm of a pericyte. Why this is the pericyte? Because you can see the basement membrane, this grayish line, here and here also. So pericytes are covered with the basement membrane from all the sides. So this is the part of the cytoplasm of a pericyte. And this is the nucleus of the endothelial cell. This endothelial cell is very busy in the process of the transcytosis. It means that it takes some substances, for example, some nutrients, and transports them inside the vesicles through its cytoplasm, releasing them into the surrounding tissue. This is why you can see a lot of rounded vesicles in its cytoplasm. It's a process of transcytosis. This is the capillary of a visceral type, or fenestrated one. Fenestrated because it has these thin areas, which are called fenestrae here. Another example of these fenestrae are in the lower picture. These arrows are pointing to the fenestrae. Through these fenestrae, large molecules can easily pass through the wall into the blood. It has no gaps, no pores, and its basement membrane is continuous. And also a part of the pericyte is seen here because it is covered with basement membrane from all the sides. This is not the whole cell, it's only a very small part of its cytoplasm. And finally, the capillary of third type, the sinusoidal type. This is the endothelium cell with its nucleus, and you can see a lot of holes, gaps and pores in the endothelium. And only in some places you can see a basement membrane. In most part of the wall of this capillary, the basement membrane is absent. So this is the sinusoidal type of the capillary. Blood vessels of microcirculatory bed, as you remember, include arterioles, precapillaries or metarterioles, capillaries, venules, and anastomoses. Arteriovenous anastomosis, through which blood can flow directly from arteriole into venule, not into capillaries. As you remember, the function of the capillaries is to perform gas exchange and also nutrients and metabolites exchange. And when the blood passes through the capillaries, in this picture its color changes from red to blue. It means that blood loses oxygen and becomes venous blood. But when the blood goes through the anastomosis, the gas exchange doesn't exist here, and the blood which enters the venule is has high amount of oxygen. Where we have arteriovenous anastomosis and why we have them. Here are two examples. We have much amount of such examples, but only two of them is here. For example, in skin, we have such anastomosis. They are needed for thermoregulation. What does it mean? If you remember the histological structure of the skin, we have the epidermis and dermis under it. And in dermis, we have so-called papillae of loose connective tissue. And in these papillae, we have very dense network of blood capillaries. The blood enters these capillaries from arterioles that are located in more deep layers of the skin. So, if the weather becomes cold, our skin starts to look more pale, whitish. Why it happens? Because the sphincter here closes and blood do not goes into the capillaries located close to the surface of our skin in the dermis papillae, but instead blood enters the anastomosis and goes directly back into the body. It drains into veins and goes in the deeper layers of our skin. So the blood doesn't go to the surface located blood capillaries. So this is why our skin will look more pale, more white. It is needed to save the heat, because when blood in capillaries come close to the surface of our body, it loses heat. If the weather is cold, our body doesn't want to lose the heat, so this is why the surface located capillaries will not be filled with blood. Blood will go not into capillaries, but from arterial into venule. And vice versa, opposite, when the weather becomes too hot, our skin becomes red. Why? Because our body wants to lose excessive heat through the surface of our body. This is why precapillary sphincters will open and blood will go from arterial to the surface located, superficially located capillaries, making our skin looking reddish to lose excessive heat. So this is about thermoregulation in skin and in kidneys. Another example of location of arteriovenous anastomosis. There, they serve to regulate the blood pressure. If the blood pressure becomes too high, in order to save the fragile capillaries in the nephrons, the excessive blood will go not into these capillaries, which can rupture if the pressure is too high, but directly from arterial to the venule to save the capillaries. Regulation of blood pressure inside the kidney. Here is the classification of anastomosis. So we have the true anastomosis, which one of them is here. True anastomosis means that the blood that goes through this anastomosis doesn't lose oxygen and goes directly into the venule. And atypical are actually the short capillaries, and gas exchange happens, but not like in the true capillaries to somewhat extent. And here is the classification of the true anastomosis based upon the histological structure of the closure apparatus, like sphincters. So this closure apparatus may consist of smooth muscle cells or some other type of cells, which is called epithelioid cells. So I think you shouldn't learn all this classification, but you should be able to say something about the types of anastomosis. Next, lymphatic system. Lymphatic system includes lymph capillaries, lymphatic vessels, and lymph nodes, and lymphatic system is connected to the blood system because large lymphatic vessels, lymphatic ducts drain lymph into large veins of the upper part of our body. So lymph finally enters the blood flow into the veins. What is lymph? Lymph is a fluid which was filtrated first from blood capillaries. So first it was the blood plasma. It went out from blood capillaries and became interstitial fluid, which surrounds cells in the tissue. And next, it was drained into lymph capillaries. Some amount of interstitial fluid returns into blood through blood capillaries or post-capillary venules, and some amount of this interstitial fluid goes into lymph capillaries and becomes lymph. Lymph contains waste products from tissues, metabolites, and also it may contain some bad substances and bad agents, for example, bacteria or tumor cells or something other bad substances. Lymph capillaries drain into lymphatic vessels. Lymphatic vessels carry lymph through lymph nodes. So lymph flows through lymph nodes where it is filtrated. It means in lymph nodes, pathogens, bacteria, tumor cells will be catched and the lymph will exit in its filtrated state. It will not be dangerous anymore. And this non-dangerous lymph finally will enter the blood circuit, into the subclavian veins. The lymph capillaries, unlike the blood capillaries, are blind closed in one end, so it has the beginning. The lymph capillary is connected to the surrounding tissue with so-called anchoring fibrils. So some amount of fluid exits the capillary, becomes extracellular fluid, and some amount of this interstitial fluid enters the lymph capillary and becomes lymph. Here you can see the difference between the structure, histological structure of the lymph capillary and the blood capillary. This is the somatic type of blood capillary. And endothelial cells of lymph capillary are much larger. This is the first difference. Larger endothelial cells. Second difference, endothelial cell in the lymph capillary has almost no basement membrane, but it is present in blood capillary. Number 3, almost no contacts between the endothelial cells in the lymph capillary, unlike blood capillary, which has all the existing types of contacts, junctions between the cells, including tight junctions. And also the pressure inside the lymph capillary is the lowest if we compare it with the pressure in the surrounding tissue and the pressure in the blood capillary. So the fluid goes from capillary to the tissue and from tissue towards lymph capillary because of the gradient of the pressure from 15 to 4 to 1 to millimeters of mercury. The structure of lymph vessels resembles the structure of the vein. The thickness of the wall is very thin, as it is in the vein, and also valves are present. This is the histological picture of the lymph vessel. It has only endothelial cells and some amount of underlying loose connective tissue. Actually, it will be the subendothelial layer with no tunica media, so loose connective tissue of subendothelial layer is continuous with loose connective tissue of adventitia. So this is the wall of the lymphatic vessel. And here are the cusps of the valve. And these actually are cross-sected muscle fibers of skeletal muscle. The lymph goes through the lymph vessel in this direction, leftward in this picture, because of the contraction of the skeletal muscles. When the muscles contract, they push the blood in this direction, leftward, and then when the muscles relax, blood tries to come back, but it enters these pockets of the valve. Valve cusps close, they come close together, and blood cannot go backwards. This is how the valves are working. The same idea is for the valves of the veins. So the blood is pushed upwards from our veins that are located, for example, in our legs, as a result of contraction of the muscles of our legs. And when the muscles are relaxed, the blood cannot go downwards as a result of the gravity because of the presence of the valves. This is how the blood goes upwards through the veins and lymph vessels, because of contraction of surrounding skeletal muscles. And our final topic for today is the heart. The heart is the muscular organ which provides the blood pressure, which makes blood to go through all the circuits. We have 2 circuits in our body, one for all the organs and second one, pulmonary, for lungs, for our blood to become rich in oxygen and to get rid of other bad substances. So the heart wall includes 3 layers: innermost endocardium, medium layer myocardium, and outermost epicardium. Endocardium, myocardium, and epicardium. The heart is located inside the pericardium, pericardial cavity filled with the fluid, and the outer wall of the pericardium is called the parietal layer of the pericardium, and the inner wall of this cavity is called visceral layer of the pericardium, which is actually the epicardium. So epicardium is the same as visceral layer of the pericardium. Development of heart. Development begins on the 70th day, and the heart develops from two germs, left one and right one. And as a result of the lateral folding, these two left and right germs become fused together to make future heart, which starts beating after 22nd day. What are these germs? Actually, endocardium develops from mesenchyme, which is located between endoderm and the visceral layer of the lateral plate mesoderm here, and the myocardium plus epicardium develops from the visceral layer of the splanchnotum, visceral layer of lateral plate mesoderm. So endocardium develops like a tube, left one and right one tube, that will fuse together to make one endocardium, and the myocardium develops from this area, and visceral layer of splanchnotum will make the visceral layer of pericardium, seen here. Visceral layer of pericardium from visceral layer of splanchnotum. This is the epicardium. And the cavity, which was previously coelom, here coelom, its outer wall, which was produced by parietal layer of splanchnotum as a result of the fusion, will make the parietal layer of the pericardium. So this will be the cavity of pericardium and visceral and parietal layers of pericardium as a result of lateral folding and fusion of lateral folds. Now, in the preparation, we will have the section through the wall of the heart, in which we can see all the three layers. This will be endocardium, this one myocardium, and epicardium. This is what you should know by heart. The structure of the heart wall. Inner layer, endocardium. It consists of 4 sub-layers. Here in the picture, this is the endocardium. It consists of endothelium, subendothelial layer, muscular elastic layer, not well seen in our preparations, and outer connective tissue layer. 4 layers. Endothelium is like in any other blood vessel, 1 layer of flat cells located upon the basement membrane. Subendothelial, outer connective tissue layers, both consist of loose connective tissue. Muscular elastic layer includes some amount of smooth muscle cells, but their amount is very, very small. They are almost not seen. They should be located somewhere in this area. So endocardium mostly consists of loose connective tissue. Next, myocardium consists of cardiac muscle tissue, which includes, as you remember, 3 types of cardiomyocytes. So this reddish in color is the myocardium. Actually, these are contractile or working cardiomyocytes. And between them, whitish areas are the layers of loose connective tissue with blood vessels and nerves. And finally, epicardium. Epicardium is in this area. It includes loose connective tissue and is covered with mesothelium, not seen in this picture. It should be the outermost layer of the heart. So heart is covered from outside with the mesothelium. Mesothelium, as you remember, like the endothelium, is one layer of flattened cells located upon the basement membrane. Now, you should know this one. What is the difference in histological sense between the epicardium and endocardium on histological preparation? Because both of them, epicardium and endocardium, consists of one layer of flattened cells, endothelium and mesothelium respectively, and underlying loose connective tissue. But the epicardium, unlike endocardium, may include large amount of adipose tissue. So here you can see adipose tissue. Adipose tissue can be present in endocardium also, but it is much more developed in epicardium. A lot of adipose tissue in epicardium. Second, large blood vessels. For example, this one. This is the large vein. Also, large arteries may be present in epicardium and not endocardium. 3. Nerve ganglia with neurons cell bodies. This structure is the nerve ganglion. Ganglion is the collection of neurons cell bodies. These are cell bodies of neurons. And bundles of nerve fibers, not seen in this picture. Another example of this preparation, endocardium, myocardium, and epicardium. In epicardium, we can see a lot of adipose tissue and large blood vessels. Another example, we shall see them in class. In this picture, at higher magnification, we can see endocardium. Endocardium includes endothelium, subendothelial layer. Here should be muscular elastic layer with elongated cell nuclei of smooth muscle cells, and outer connective tissue layer. And here the myocardium starts. Another example, large vein in the epicardium, and intramural parasympathetic nerve ganglion. Intramural means located inside the organ, inside the wall of some organ. In our example, inside the heart. Parasympathetic means about parasympathetic nerve system. Ganglion means the collection of cell bodies of neurons. These violet rounded structures are neurons. These blue dots are cell nuclei of glial cells, as you remember them, supporting cells in nervous tissues, and also cell nuclei of cells in loose connective tissue. Another example of nerve ganglion, group of cell bodies of neurons, one neuron, another one, another one, another one. All the dots are cell nuclei of glial cells and loose connective tissue. Intramural parasympathetic nerve ganglion. Ganglion, and here a bundle of nerve fibers. It consists of nerve fibers, and it's a little bit wavy. The bundles of nerve fibers are always a little bit wavy in our preparations. And the cell nuclei that we can see here are the nuclei of glial cells, Schwann cells. Another example of a bundle of nerve fibers in epicardium, adipose tissue, loose connective tissue, and mesothelium. This is the myocardium, consisting of working cardiomyocytes. And for class, when you will have your practical class, you should repeat all the information about the muscle tissues. about especially the cardiac muscle tissue, all that you know about it. So these are the working cardiomyocytes with lateral anastomosis, with intercalated discs somewhere here, with centrally located cell nuclei, and so on. Intercalated disc, lateral anastomosis, centrally located cell nucleus. Cross-sected working cardiomyocytes, intercalated disc and its structure in the transmissional electronogram. It's a group of contacts, including gap junction or nexus, desmosomes, here and here also myofibrils in both cardiomyocytes, mitochondria, and so on. You should also remember all that you know about the types of cardiomyocytes. We have typical one, secretory, and atypical. Most of the cardiomyocytes in our heart are contractile or working or typical cardiomyocytes. They are for contraction. A secretory one is for production of natriuretic factor, and it is located in the atria of the heart, not in the ventricles. And atypical are for production and conducting of electrical impulses. Secretory cardiomyocyte. This is the part of its cytoplasm with secretory granules with atrial natriuretic factor. How it works? If the blood pressure, BP is blood pressure, if the blood pressure becomes too high, it is not good for heart, and heart wants this blood pressure to be decreased. So secretory cardiomyocytes release their atrial natriuretic factor into blood. This factor goes to kidneys and acts upon the tubules of nephrons, making them to release sodium and water with urine from our body. So our body loses water with urine. It means that the amount of water in our blood decreases. It means that volume of blood decreases. If the volume of our blood in our blood vessels decreases, it leads to decrease of blood pressure. This is how it works. Another target cell of this atrial natriuretic factor is the smooth muscle cell of the arteriole. When this chemical acts upon the smooth muscle cell of the arteriole, it makes this cell to relax. It leads to increase of the lumen of the arterioles, and it leads to decrease of blood pressure. So double acting of this chemical atrial natriuretic factor from secretory cardiomyocyte makes blood pressure to be decreased. Now, finally, cardiac conduction system is for generation, conduction of electrical impulse, and as a result, for making working cardiomyocyte to contract. These cells include three types of atypical cardiomyocytes. First, pacemaker cells, second, transitional, third, conducting ones. Pacemaker cells are located in nodes, sinatral node, atrioventricular node, and they serve for creation of electrical impulses, about 60 per minute. Transitional ones, also located in nodes, they transmit this electrical impulse from pacemaker to conducting cell. And conducting cells, making elongated structures named His and Purkinje fibers. They consist of conducting cardiomyocytes, and they conduct electrical impulse from these conducting cells to working cardiomyocyte to make them to contract. This is how these conducting cardiomyocytes look like in our histological section. This is the picture of it. Endocardium, myocardium consisting of working cardiomyocytes, and this is the so-called Purkinje fiber. This is the elongated structure consisting of several conducting cardiomyocytes. They are elongated with scattered myofibrils in the periphery, and they are rounded, unlike elongated working cardiomyocytes. And this is our real preparation that you will see in our class, and you will make, I think, some picture of it. Purkinje fibers located between endocardium and myocardium. The high magnification endocardium, myocardium, Purkinje fiber consisting of several conducting cardiomyocytes, oval or rounded, with large cell nucleus. This is how it looks like on electronogram, nucleus, and scattered cross-sected myofibrils at the periphery. And final picture in your album will be the valve of the heart. Valves allow blood to flow in one direction, for example, from the atrium to the ventricle, and prevent the backflow because they are connected. These cusps of the valves are connected to the walls of the atrium with elongated chordae tendineae, which prevent these valves from inverting. This will be explained in class, but some additional information for you. So this is the cross section through the wall in the area of the cusp of the valve. This is the cusp of the valve. In the center, we have dense fibrous connective tissue, which is attached to the skeleton of the heart, which is located between the atrium and ventricle. So all the other information will be given you in class. So if you have any questions, you may ask them in email or using WhatsApp. So glad to see you, to hear you. See you in class.
Thank you, Professor.